Hello everyone, I am Richa and my topic for today's discussion is ROS production in ETC. So first of all, what are ROS? ROS are reactive oxygen species that are highly reactive chemicals formed from oxygen. They can also be defined as the byproducts of the normal metabolism of oxygen. Utilization of molecular oxygen by aerobic organisms inevitably results in the formation of a number of oxygen containing reactive species. Those are collectively known as ROS. ROS are the type of unstable molecule that contains oxygen and that are easily reacts with other molecules in a cell. The ROS is derived from both endogenous and exogenous sources. Some of the endogenous sources are mitochondria, peroxisomes, endoplasmic reticulum, phagocytic cells, and the exogenous sources are pollution, alcohol, tobacco smoke, heavy metals, etc. ROS play a twofold job as both beneficial and toxic component to living system. At moderate and low levels, ROS has beneficial effects and involve in various physiological functions. But at higher concentration, ROS cause oxidative stress. The oxidative stress is developed when there is an excess production of ROS. Every biological component has its negative and positive effects. So the negative effects of ROS contains uh, it damages DNA and RNA, oxidation of polyunsaturated fatty acids in lipids, which causes lipid peroxidation, oxidation of amino acids in proteins, oxidative deactivation of specific enzymes by oxidation of cofactors. It also causes oxidative damage. And lastly, impairment of cognitive function is accompanied by an accumulation of oxidative damage. There are also some of the positive role that is played by ROS. ROS is critical and essential for memory function. It gives defense against of pathogenic microorganisms, and it plays a positive role in mitogenic response and in oxidate redox reaction. Now, coming to the ROS production through ETC. So, first of all, what is ETC? ETC is known as the electron transfer chain. It is a series of protein complexes that couple redox reaction, creating an electrochemical gradient that leads to the creation of ATP by oxidative phosphorylation. It occurs in mitochondria in both cellular respiration and as well as photosynthesis. The chemiosmatic theory proposed by Peter Michel in 1961 states that the transfer of an electron derived from the substrate oxidation and ATP synthesis are coupled in a mitochondrial ETC. The mammalian mitochondrial electron transport chain includes complex 1 to complex 4 as well as an electron transport ubiquinone and cytochrome C. There are two electron transport pathways in ETC. Complex 1, 3 and 4 with NADH as substrate and complex 2, 3 and 4 with succinic acid as the substrate. The electron flow in an ETC is coupled with the generation of a photon gradient across the inner membrane, and the energy accumulated in the proton gradient is used by complex 5 to produce ATP. Mitochondria are a main source of cellular respiration. Generation of ROS mainly takes place at the electron transport chain located on the inner mitochondrial membrane during the process of oxidative phosphorylation. Leakage of electrons at complex 1 and complex 3 from electron transport chain leads to partial reduction of oxygen to form superoxide. Superoxide is quickly dismutated to hydrogen peroxide by two dismutases, including superoxide dismutase 2 also known as SOD2 in mitochondrial matrix, and superoxide dismutase 1, also known as SOD1 in mitochondrial intermembrane space. Both superoxide and hydrogen peroxide generated in this process is called mitochondrial ROS. Generation of electron leaks and proton leaks in the electron transport chain. Electrons derived from oxidizable substrate are passed through complex 1,3,4 or complex 2,3,4 in an exergonic process that derives the proton pumping. The site of ROS production in each complex are indicated, including the sites 1F or 1Q in C complex 1, 2F in complex 2, sites 3Q0 in complex 3. The oxygen released into the intermembrane space by site 3Q0 can be converted into hydrogen peroxide in a reaction catalyzed by superoxide dismutase 1 or SOD1. Hydrogen peroxide then may diffuse into the cytoplasm. The red arrows indicate the electron pathways in the given diagram, and the black arrows shows the substrate reaction. ROS can be generated in the matrix at both sites, uh, known as 1F or FMN site and 1Q or Q0Q binding site during the transfer of electron from NAD to Q0Q in C1. In complex 1, which is also known as C1, also known as NADH ubiquinone oxidase, is the largest multi-subunit enzyme complex in the ETC. The key role of C1 in the transfer of electron from matrix to NADH to ubiquinone. Complex 2 namely succinate dehydrogenase, is a component of the Krebs cycle as well as the ETC. FAD is reduced to FADH2 after receiving electrons from succinate and then transfers the electron to FES clusters. Then CQ0Q is reduced to QH2. Coming to complex 3. In complex 3, which is also referred to as cytochrome B-C1 complex or CQ0Q cytochrome C reductase, and transfers the electron carried by QH2 to cytochrome C. The electron transfer process is accomplished by the Q cycle. Complex 4, also known as cytochrome C oxidase, transfers electron from cytochrome C to the terminal electron acceptor or oxygen to generate H2O. The last complex, or also known as the ATP synthesis complex 5, is normally called as F1 F0 ATP synthesis and consists of two functional domains, F0 and F1. The ETC transfers two electrons at a time to monooxygenase to generate one H2O molecule along with 4, 2 and 2 electrons found from complex 1, 3 and 4 respectively. Then the protons pass from the intermembrane space to the matrix through F0, which transfers the stored energy created by the proton electrochemical gradient to F1, causing conformational changes so that ADP can be phosphorylated to form ATP. So this is how the ROS production occurs through electron transport chain in mitochondria. That was all about my topic. Thank you.